All right, in this video, we will conclude Unit 6 by talking about chromosomal errors that can happen as a result of meiosis. And one of those chromosomal errors is called nondisjunction. The easy definition for nondisjunction is a failure for chromosomes to separate at some point during meiosis. We know that in meiosis 1, homologous pairs separate. In meiosis 2, sister chromatids separate. If at some point along the line there is a failure to separate, then the resulting gametes will have extra chromosomes or will have fewer chromosomes than they should have. Let's look at this picture to see this. And so up here in meiosis 1, this is the correct version of this, right? Homologous pairs should separate, but let's say that they don't. This cell over here is going to get both of these. This cell over here is going to get none of those. And so then the resultant, and then so they divide normally in meiosis 2. And so here you have a gamete that we'll call N plus 1. N standing for haploid plus 1 in that it has an extra chromosome. Here are N minus 1 cells because they have one less chromosome than they should. And so this, when nondisjunction fails in meiosis 1, you have two N plus 1s and two N minus 1s. Notice here in this side, nondisjunction occurs in meiosis 2, where sister chromatids fail to separate. And so one cell has N plus 1, the other has N minus 1, but over here, these separate normally, and so these are just called N. And so nondisjunction can occur in either meiosis 1 or meiosis 2. This is how you get a disorder like trisomy 21 or what we call Down syndrome, because this individual has 3 21st chromosomes. Either mom or dad had too many chromosomes, had an N plus 1 gamete basically on their 21st pair. And so when that gamete combined with the other gamete, you get too many. A lot of times the resulting zygote from this sort of fertilization event will just spontaneously die because it's not viable. But for certain individuals, this can result in a viable offspring, but just having some issues associated with that, having that extra chromosome. And so we all have met people with Down syndrome, and they have a variety of symptoms as a result of that. But so it just results from that nondisjunction. There's nondisjunction on other pairs, but trisomy 21 is usually the one that you're going to see when it comes to like a test question or something like that, just talking about nondisjunction. But the big thing is understanding how this occurs here, and how if nondisjunction occurs in meiosis 1, there's one result, and if it occurs in meiosis 2, there's this whole other result. There can be results, or there can be errors on the chromosome structure itself. And so you can see there on the left just the words, and they all mean things. So deletion, this is when a section of a chromosome is deleted. You may have multiple genes that are deleted here. Duplication, a section of a chromosome is duplicated. Inversion, a section of a chromosome is literally turned backwards. Instead of BCD, you get DCB. And remember, DNA is read a certain way, so this could be bad. And then translocation, where a portion of one chromosome relocates itself or translocates to a portion of another chromosome. These are typically all bad things, and they result from a faulty crossover. It's not necessarily bad, though. These kinds of mutations can lead to favorable adaptations, as natural selection has demonstrated, but most of the time this leads in a inviable type of cell. And then the last thing is about extranuclear DNA. Remember, there's DNA that is found inside the mitochondria, and mitochondrial DNA is always inherited from the mother because the mother brings the egg, and the egg has the mitochondria. The sperm cell does not donate any of its mitochondria. And so any sort of disease that's associated with extranuclear DNA or mitochondrial DNA is inherited directly from the mother. There's one in particular called mitochondrial myopathy, where the mitochondria lack the ability to make large quantities of ATP because they possess some sort of faulty DNA in them, and those proton pump that exist inside the mitochondria don't do right. They're not working correctly, so cellular respiration doesn't happen, and so they don't produce enough ATP. The general symptom of this is that the individual is just tired all the time. It's hard to get energy, but this comes from the mother's mitochondrial DNA.